This video should give you a good overview of the molecular biology suite in the LabThread system. Now, as you can see here, we have a plasmid that is loaded. You could either load it via a GenBank file or a FASTA file or a .dna, a SnapGene file. All of those will work. And if you wanted to add a new one, you would click plus plasmid, drop your file into here, and then click skip create, and then you'll immediately load that plasmid. Now we have this one loaded here at the moment, and you can see it has, you can scroll, you can zoom by holding control if you need to. You can change to a linear view, and by holding control and shift you can zoom in on that one as well. If you want to turn off both the linear and the circular view, you can do that, so you can just look at the sequence on its own. Now if we'll turn back on the circular view, just for simplicity. Now there are various different things you can do within here. So for example, you could click this region here, you could click Tools, Design Primer Pair, say it's an exact target, find primers, it'll find the primers automatically. Obviously picked a sequence with a particularly bad feature, so you can click and see what's wrong with those primers. I'm just going to add them anyway. It's now added that primer forward, that primer reverse. If I wanted to add tails to those primers, I would double click it, add the tail in there. If it's phosphorylated, you can say that it's phosphorylated as well, and you'll see that a little P will appear as well as the tail that I have just added. Other things that you can do, if you want to click on a feature to see how it's been aligned, you can double click it, show you exactly how it's been aligned. If you want to change the color, you can do that as well. If you wanted to, for example, code and optimize a gene, you could click this gene here, I could click Tools. If I wanted to just assess the homology within that particular region of DNA, I could do that. If I wanted to remove restriction sites just from that piece of DNA, I can do that. If I wanted to reverse complement it, I can also look at code and optimization. So here it's now, actually it's already optimized it, it's very fast. If I wanted to change the species, I can do that as well. And then I can also detect different repeats within the sequence. For example here, homopolymers, direct repeats, inverted repeats. If I then want to remove restriction sites and polyadenylation signals as well as splice sites, I can do that. You'll see here if these two homopolymers, if I click remove repeats, it's automatically removed them from the sequence down here. And then this button that you can't see behind me, I would then be able to replace the sequence in the plasmid map, and you can see here all of the features of the new sequence versus the original. Other things that you can do, you can annotate sequences with comments, so you can add, say, create comment, for example, create comment. You can turn it into a design, right? So you can extract that as a linear DNA, for example, or a new plasmid, if that made sense. Turn it into a primer or turn it into a feature as well. If you want to scan for features from within your database, the system comes pre-populated with many different common features. You can click scan features. Again, it's very fast. It'll do that almost instantaneously. You can turn them off. So if you don't want to import, for example, those particular types, you can do that. And if you want to review the newly recognized ones compared to the ones that are already connected, you can do that using these tabs here as well. You can turn on the enzyme settings using this little toggle here, this little drop-down menu here. You can create your own lists as well if you want to. That's the subject of a different video. You can then turn on whatever types of enzymes that you want. You can then filter them like so. And then if you wanted to lock them in, as I have done this top one, you can lock those enzymes in as well. If you want to learn more about those enzymes, you can click on them as well. It'll load out to the right-hand side. You can look at things like the buffer reactivity. Obviously, this one is only made by this company, the buffer reactivity. You look at any isoschismers or enzymes with the same overhangs as well as its properties. Now, one of the benefits of, before I go on to anything else, so translations, you can also look at open reading frame detection. You can turn on the different frames of the amino acid sequence as well. And then features, you can toggle those on as well by just toggling them on and off accordingly, or by type if you need to as well. If you've got primers, you'll be able to see those listed all here separately as a primer list. And if anybody has annotated comments, you'll be able to click and see that as well. Now, the main benefit of the LabThread system is it doesn't just sit as a standalone cloning program. So although you can edit your sequence within here, you can collaborate with your colleagues. So you can click Options, Share, select the other account within your LabThread system that you want to work with you collaboratively on this design. You can give them viewer rights, edit rights, or co-owner. If you give them the co-owner rights, that allows them to do everything that you can, so they'll be able to share it with additional people as well as delete and archive the design. Editor just allows them to edit the sequence and add annotations and things like that. Everything that's done within the system is captured within the audit log here, so you can see everything that happens is fully recorded. If you want to do a gel simulation, you can do that. Click Create Gel. You can then create your own gel simulations. You can define your own ladders in the setup area. You can set your own defaults as well. If you wanted to do sequence alignment, you can do that by clicking Create Sequence Alignment. You can then select the alignment algorithm that you want to use, upload as many files as you need to align, and it will automatically align the sequence to your plasmid map. As a reference, if you had two designs in the system that you wanted to compare, you can do that by clicking Add Design, so it allows you to compare two different plasmids at the same time. The other benefit of the fact that this is a unified system is that you can log plasmids of this design. So, for example, here if I click Log Plasmids, I could log 5 plasmids, log them as the same type of design. It automatically populates that. It automatically predicts what the tube will be. Again, you can control that yourself. You can add things like the concentration here, transfer down. We've made it as easy as we possibly can to populate this system. And if you want to say where those tubes live, you can skip straight to storage assignment. Say it lives in this box. Transfer that down. I don't know what the free slots are, so I click that and it'll automatically show me what the free slots are. And then I could log those samples. To see what samples exist of that design, you can click on this Samples tab. This will show you any samples within the system. If you wanted to understand where these samples have then subsequently gone, for example, if I click on this one, I can then click on its lineage. That will allow me to then see where that plasmid has subsequently gone to. So for example here, I've just made up some data. It's gone into a lentiviral vector sample. I can then expand that and I can see that that has gone into some HEC293 cell line clones. So this just gives you an idea of how samples are being used within the system, but they all link back to the original design that you created within the LabThread system. Now, if you want to understand where this particular sequence has been referenced, i.e. in an electronic lab notebook entry or in a particular plasmid, a particular project, you can do that by clicking on tasks. Click on tasks, it'll show you any projects where it has been referenced and any tasks that it's been used in. So those are some of the main features of the molecular biology suite. And then finally, just to show you that you can also ligate DNA. So you can select two enzymes. I didn't select two enzymes there, sorry, 1 second. Select two enzymes, right click, add to ligation. There we go. Give your ligation a name. And then you could add multiple fragments into that ligation. There is a separate video showing how the ligation functionality works, so I won't cover it in detail here. There'll also be a Gibson assembly tool being added in the next few days. For example, here, Gibson assembly is currently grayed out, but you'll be able to use that probably by the end of July 2026. So I hope this gives you a bit of background on the various different things that you can do with the plasmid system or the genetic sequence viewer within the LabThread system. And before I go, I've just remembered another really nice feature, which I'm just going to highlight. If I click on this particular location, I can click on Tools, Genome Browser. You can then select the genome that you want to navigate to. I'm just going to leave it at that one. I'm going to then type in a particular gene. Find that gene. This is not our system being slightly slow. It is the IGV. It's saying that it couldn't find it, but it has actually found it. I'm going to click on that gene. I'm going to then insert the gene into the design. It is now determined where it's going to insert it, and you can see here it has inserted it with all of the exons and introns fully labeled from the IGV browser. So that allows you to access genes that are in the public repositories and insert them into your plasmid designs as well. So I forgot about that feature, so apologies, but hopefully this gives you an overview of how the molecular biology suite works within the LabThread system. We are adding new features all the time. Do please let us know if there's something that you want us to improve or an additional feature that you want us to add, but we hope that you find this a very useful tool for your molecular biology needs. Thank you for visiting LabThread.